Hi everybody, Dr. Mike here. In this video we're going to go through beta-lactam antibiotics. We're going to go through their mechanism of action, and we're also going to have a look at antibiotic resistance to beta-lactam antibiotics. So, first thing we need to look at is the class of beta-lactams. So there's three types of antibiotics I want you to remember that sit underneath this category. They include the penicillins, they include the cephalosporins, and the carbapenems. Now these antibiotics are called beta-lactam class antibiotics because they contain something called beta-lactam. And beta-lactam looks like this. Looks like a little house. Now that's the chemical structure of it, and it plays a really important role in killing off bacteria. What type of bacteria? Well, for penicillins, mainly gram-positive, cephalosporins, positive and negative, and for carbapenems, positive and negative as well. Now, I've drawn up a stereotypical bacteria here. It's neither positive or negative. You could make an argument that it's gram-positive because gram-negative bacteria have another cell membrane on the outside of a very thin cell wall. So, what we've got here is a cell membrane on the inside, similar to ours, phospholipid bilayer, except for this bacteria, like most bacteria, they do not contain sterols. Sterols help maintain the rigidity and structure of the cell membrane, which means bacterial cell membranes aren't that strong. Remember that point. Next thing is you can see a cell wall. Now, we do not have cell walls, which means this makes it really important to exploit when we want to kill off bacteria. We can target something they have that we don't to kill them off and not kill us off. And that's how these beta-lactams work. They target this cell wall. But how? Well, let's see what the cell wall is made up of. You can see that there's three colors I've put here. You can see that I've got a red color, I've got a blue color, and I've got a green color. Let's do the green last. Now, what this blue color is, well, both of these, what they are basically sugars. They're glycans. And what they are made up of is the blue one is known as N-acetyl glucosamine, and the red one is N-acetyl muramic acid, NAG and NAM. Now, you can see attached to NAM is the green thing. The green thing is amino acids, and usually there's around about 1, 2, 3, 4, 5 amino acids attached to it. Now you can see that the amino acids are bound to the amino acids of the next layer, so they need to be clicked together. Now this is where it becomes really important. In order to click these together, so I'm going to draw up another NAG, I'm going to draw up another NAM, there's the sugars, and I'm going to draw up the amino acids attached to the NAM, 2, 3, 4, 5. Now in order for them to click together, we need something really important. This is called a transpeptidase. Transpeptidase looks a little bit like that. And what the transpeptidase does is it comes on and clicks the amino acid groups together, allowing for a nice cell wall to be produced. The cell wall maintains the rigid, strong structure of the bacteria. Okay, this is now where the beta-lactam ring comes into play. We've got the beta-lactam ring here, which is common amongst these three types of antibiotics. When we give individuals these antibiotics and they are infected by particular bacteria, when this bacteria starts to make its cell wall, it needs this transpeptidase. Let's actually write that down, transpeptidase. And there's different types. The type of transpeptidase we need to talk about is called penicillin binding protein. This is the type of transpeptidase we need to talk about. Here it is, PBP. All right. Beta-lactam comes along. It binds into the transpeptidase and it stops it from being able to cross-link the amino acids together. This stops the cell wall from being made, and that means the cell wall peels away and all that we are left with is the cell membrane. And I told you for bacteria, they don't contain sterols and not a very strong rigid structure, and this then means that fluid can rush into the cell. Why would it rush into the cell? Because bacteria are hyperosmotic. a high concentration of stuff inside compared to outside, so water wants to rush in. If water rushes in, the cell, bacterial cell, gets big, fat, and round, and it bursts. All right, this is how these particular antibiotics work. All right, next thing is this: how do they become resistant? Now, how does the bacteria become resistant to these types of antibiotics? Well, what happens is it can happen two ways. First way is this: when we look at the DNA inside of a bacteria, it is circular DNA, it is double-stranded DNA. But they still contain nucleotides. Those nucleotides still create genes. So remember, genes are the smallest portion of DNA sequences that can be turned into proteins, and proteins have a function. All right. Now let's just say one of these genes turns into the protein that is transpeptidase, specifically penicillin binding protein. All right. Let's say that there is a mutation in this particular gene. And what this mutation does is it changes the gene to encode a transpeptidase that's shaped slightly different. Instead of having a transpeptidase that looks like that, maybe the transpeptidase looks like this. It can still have its function of cross-linking the amino acids, but it means that the beta-lactam ring, is it going to be able to bind in there? The answer to that is no, it will not. So these antibiotics no longer work when this bacteria has the mutation within the gene that encodes transpeptidase. So that's one way that resistance can occur. The next way that resistance can occur is that other bacteria, let's just say I've got another bacteria here, and let's just say that this bacteria, for its own DNA, has a gene, and this gene encodes an enzyme that can actually break down beta-lactam. So let's just say it has a gene here that encodes for a protein that can break down beta-lactam. What you probably don't know about bacteria is that when they come together, they can share genetic material. And let's just say that these two bacteria share their genetic material, and this gene now can be inserted into this particular bacteria. And now it produces an enzyme called beta-lactamase. And this beta-lactamase likes to break down beta-lactam. And if it's breaking down beta-lactam, it means, again, it no longer works, no longer fits within the transpeptidase, no longer stops that bacterial wall from being synthesized. So, what have we gone through? We've gone through the fact that beta-lactams, there's three main types of antibiotics that sit within this class: penicillins, cephalosporins, and carbapenems. The beta-lactam ring stops the transpeptidase from linking the amino acids together within the peptidoglycan cell wall. This means the wall falls apart, water rushes in and bursts the bacterial cell. The way that bacteria can become resistant to these types of antibiotics: two ways. One, it gets a mutation within the transpeptidase itself and changes its shape so the beta-lactam no longer works. The second way is that it can get a gene through another bacteria that encodes an enzyme called beta-lactamase. The ASE on the end means it's an enzyme that breaks down the beta-lactam. Let's label this as beta-lactamase. It breaks down the beta-lactam and stops it from working. So this is a quick run through of the beta-lactams.